Hello students, how are you all? Today in this lecture I am going to explain about the embryo culture and what are the different methods of embryo culture. Why we have to do embryo culture? Because in some species we can observe about the embryo abortion. Then why this embryo abortion will happen and what it is? Embryo abortion means that is premature termination of the embryonic development in plants. And why this embryo abortion will happen? This is because of pre-fertilization barriers and post-fertilization barriers. What are the different pre-fertilization barriers that can capable of leading to embryo abortion? First one is differences in flowering time of two parents, that is in male parent and female parent. If we will see, the flowering time used to be different, and lack of stigma receptivity and pollen viability. In female parent there is a lack of stigma receptivity even though pollen is viable, or if stigma is receptive but pollen is not viable, then also it lead to pre-fertilization barrier and inability of the pollen to germinate. Even though pollen is viable and stigma is receptive, but after falling the pollen on stigma, it is unable to germinate. That can also lead to the embryo abortion. And failure of pollen tube to reach the ovule due to slow growth of the pollen tube. And cross incompatibility due to any other reason. Cross incompatibility, if we are doing the crosses between the two different genus or two different species which are not at all compatible to each other, that will also lead to the embryo abortion. And coming to the post fertilization barriers, the post fertilization, the name itself indicating that these barriers will happen after the fertilization finished, that it will hinder or retard the development of zygote after fertilization. This may be due to degeneration of endosperm that leading to starvation and abortion of the hybrid embryo. This endosperm is the main nutritive tissue that will provide the nutrients to the embryo development. And embryo endosperm incompatibility, in such case the endosperm will release some chemical substances which are toxic to the embryo and that kill the embryo. And then third reason may be malfunctioning of the endosperm due to abnormal behavior of other cells like antipodals, nucellus, integuments which surrounding the embryo. These are the pre-fertilization barriers and post-fertilization barriers that can lead to the embryo abortion. So, how to overcome these barriers? These pre-fertilization barriers can be overcome by in vitro pollination and in vitro fertilization, whereas post-fertilization barriers can be resolved by excising and culturing of young hybrid embryos. Why I am calling these hybrid embryos means these are generated because of intergeneric and interspecific crosses. So, these can be grown by embryo culture, embryo implantation, ovule culture and ovary culture. So, I will discuss about each one by one. What is embryo culture? Embryo culture deals with the sterile isolation and in vitro growth of mature or immature embryo with the ultimate objective to obtain a viable plant. That embryo culture conventionally it means sexually produced zygotic embryo culture. How the embryo culture exactly started? Layback during the year 1925, he isolated the interspecific hybrid embryos of Linum perenne and Linum austriacum. He tried to grow the seed which was produced from this cross, but he was unable to grow that seed. So, then what he did, he isolated the embryos and he grew on moist filter paper or cotton soaked in 15% glucose solution. Then he could able to provide the viable plant. From the time this embryo culture was exactly started. So, there are two types of embryo cultures. One is the mature embryo culture and second one is the embryo rescue, which is also known as immature embryo culture. So, let us see one by one. What is mature embryo culture? The mature embryo, the name itself indicating that the embryo is already matured. That is, we are isolating from ripe seeds and cultured in vitro. Mature embryo cultures are carried out in the different following conditions. When the embryo remain dormant for long periods, that is, it is unable to germinate, and low survival of embryos in vivo, when it is inside the embryo sac, it is unable to survive, and to avoid the inhibitors present in the seed for germination. Inhibitors means whatever chemical substances present in the seed coat or endosperm or cotyledons, they will inhibit the germination of the seed. And the seed dormancy also, that may be due to the seed coat imposed dormancy or environmental dormancy, but here we can overcome the seed coat imposed dormancy because of this mature embryo culture, and for converting sterile seeds into the viable seedlings. So this seed dormancy may be due to chemical inhibitors or mechanical resistance that exerted by the structures which are covering the embryo, generally that is integuments. So that can be successfully bypassed through the embryo culture. And mature embryo culture is relatively very easy because the embryo is already matured, and we can grow these mature embryos on a simple inorganic medium which is supplemented with the energy source. Generally energy source we will use the sucrose, and it is possible because the mature embryos are excised from the developing seeds which are in the autotrophic in nature. That means they can capable of synthesizing their own nutrients. This is the mature embryo culture. Now let us see about the immature embryo culture, which is also known as embryo rescue technique. The name itself indicating that immature embryos. Immature embryo means it is not yet fully developed. So embryo rescue refers to a number of in vitro techniques whose purpose is to promote the development of immature or weak embryo into a viable plant. And this approach where we will use means to avoid the embryo abortion and to produce a viable plant. And this is widely used for producing plants from hybridization, in which the failure of proper endosperm development causes the embryo abortion. Because here in hybridization experiments where endosperm used to be failed, when it will not form, then who will provide the nutrients to the embryo, because endosperm is the main structure that is responsible for providing food material to the growing embryo. So here endosperm is not there, then embryo abortion will automatically happen. In embryo rescue procedure, the artificial nutrient medium which we are providing, it will serve as a substitute to the endosperm and thereby allowing the embryo to grow in the normal. And most important application of embryo rescue is production of interspecific and intergeneric hybrids from the wild plant species which do not normally produce the viable seed, that is between interspecific and intergeneric hybrids. There is a lot of disturbance from the background side because lot of construction work is going here, so sorry for that, but anyhow I will try to give more clarity in this video. So what is the important aspects of the embryo culture? There are two aspects. First we have to exercise the embryo, and second one, what cultural requirements, then what are the nutrients required by the embryo to grow into a viable plant. So first we will see the cultural requirements, that is also known as factors involved in the embryo rescue. First most important one is the nutritional requirement, that is generally in embryo growth there are two phases, one is heterotrophic phase and second one is the autotrophic phase. What is heterotrophic phase? Heterotrophic phase is the early phase of the embryo which is mostly dependent on the endosperm and maternal tissues for the nutrient supply, and second one is the autotrophic phase. That phase is characterized by the metabolic capability of the embryo to synthesize substances required for the growth and which slowly makes it independent. These two phases are okay, but the intervening phase used to be there, that is conversion from the heterotrophic to autotrophic phase, that is very most important. and nutrient supply also used to be vary according to that embryo which is used to be in which phase, that is whether it is a heterotrophic phase or whether it is a autotrophic phase. So the composition of medium for culturing the immature embryos. Immature embryos means they require a lot of nutrients because the embryo is not yet matured, so the nutrient medium used to be highly complex, whereas in case of mature embryos it is simple media. And the transfer of embryos from one medium to another medium while growing the immature embryos is essential because for each stage nutrient requirement will be different. So let us see composition of the medium, what should be there in the medium. So inorganic constituents, generally we will use Murashag and Skoog media, which is commonly known as MS media, and Gamberg's B5 media, that is GB5 media, and white media. These are the three media generally we will use to raise the embryos. And the most commonly energy source is the sucrose, and the nitrogen source will be the ammonium nitrate, and for various amino acids we will use the casein hydrolysate, and natural plant extracts like coconut milk, that is liquid endosperm of the coconut milk, and the pH range we will maintain as a 5 to 7.5 and temperature 24 to 26 degrees centigrade. These are the ideal conditions where I am listing out. And better growth of embryos we can first we can see in the darkness, then later after germination we will transfer these embryos to the light. And during the culture conditions embryos are grown into a plantlet. After growing into a plantlet, then we will transfer them to the sterile soil. That is, first we have to give seedling hardiness. Then if we will see the individually what are the things we have to provide for the embryo growth. First one is the carbohydrate. Sucrose is the best carbon source which produces the energy and also maintains suitable osmolarity, because osmolarity has to be maintained to prevent the precocious germination of the embryos. Mature embryos grows well with the 2% sucrose, but younger embryos need high level of sucrose concentration, that is 8 to 12%, and mannitol at 120 grams per liter is also suitable for heart shaped embryos. Coming to the amino acids and vitamins, in general nitrogen we will provide in the form of glutamine 400 milligrams per liter, and asparagine which is beneficial for younger embryos, that is immature embryos, and casein hydrolysate, already I told that especially in case of citrus we will use, and ammonium the medium, that is essential for the proper growth and differentiation of the embryo. These are the amino acids and vitamins. Then what are the mineral salts we have to provide? MS medium, that medium favors the higher survival rate of cultured immature embryos and also supplemented with minerals like potassium, calcium, and reduced level of ammonia nitrate and iron EDTA. Then natural plant products, generally we will use the liquid endosperm of the coconut, that water we will use. This one first recommended by the Van Weverbeek during the year 1941. And why we have to use this liquid endosperm of coconut? I think you know that these are rich in cytokinins, and they told that this may be due to presence of some endosperm factors in liquid endosperm of the coconut. In some tissue, this rising of these embryo cultures, they will use extracts of the dates, bananas, tomato juice, and wheat gluten hydrolysate. They found that it will promote the growth of exercised embryos of the barley. Growth regulators, generally they will not use auxins and cytokinins in case of mature embryos, because already they matured. In case of immature embryos, if they required, then according to the stage we have to use these growth regulators. And pH of the medium, general pH already I told that 5 to 7.5, and these pH media it will vary according to the different crop what we are using. Like in case of barley, if you will see 4.9 to 5.2, rice 5 to 9, and datura 5 to 7.5. These pH conditions are standardized based on researchers who did experiments with these crops. Culture environment. Culture environment means what is the temperature you are maintaining, that is generally 24 to 26, and it will vary according to the species to species, and it ranging from 25 to 30 degrees centigrade. And light does not appear to be critical in several cases in case of barley, but it suppresses precocious germination. Initially we have to grow these embryos under the dark condition, later we have to transfer to the light, and after growing of the seedling then we will transfer finally to the soil, right. Then temperature and light. The growth requirement of embryo that often mimic those of their parents, because embryos of cool season crops they require lower temperature, whereas warm season crops they require higher temperature. And cultures are usually initially cultured in the dark to prevent the precocious germination, then later moved to the lightened environment to allow the chlorophyll development, that is after 1 or 2 weeks in the dark. Then time of culture. Time of culture is also very important to get success in this embryo culture, because cultures are often initiated at various intervals following the pollination to maximize chances of recovering the viable plants. That means after pollination, that based on the species from which we are going to grow the embryos. So we can start at any time, that depends upon the species. That interaction between the media and the time of culture is expected. It is important to test a range of media ranging from complex with high sucrose to simple with low sucrose at various culture times. So how we can come conclusion that each species require this amount of the minerals, this amount of the carbohydrates, means first we have to standardize each one. That means what are the mineral things we have to add. If we are adding excess, what is going to happen? If we are adding low amount, then what is going to happen? So the scientists used to do research based on these aspects, and finally they will come to the conclusion that this amount is good for grow the embryo from this particular species. And what is the general embryo rescue process? That is excise the embryo. Then most commonly used embryo rescue procedure is embryos are excised and directly they placed on the culture media. And here embryos generally used to present in the sterile environment. Some people suggest that there is no need due to sterilization of these embryos, because they are already present in the embryo sac and they are in the sterile environment. Then fruit from the controlled pollination of greenhouse. In case of valued crops, we used to do pollination in the greenhouse or glass house conditions, or field grown plants that is directly collected prior to the time in which we are thinking that embryo abortion is to be occur. Then embryos are located in a sterile environment. Dissection of the embryo itself is not required, but in some cases entire ovary is surface sterilized before excising the embryo. Then excised embryos directly placed into the culture after excision, and it does not become dry, because culture medium is already containing the nutrients and water, everything is required for the embryo growth, because this culture act as an alternative to the endosperm for providing nutrients to this embryo. In case of hot shaped and younger embryos, the embryo should be excised with the suspensor intact, because suspensor has to be provided. It acts as an anchor to the embryo to absorb the nutrients from this culture medium. That is why in case of immature embryos, we have to excise the embryo along with the suspensor. Then second one is the embryo endosperm transplant. What is this embryo endosperm transplant? Means isolation of immature embryos often poses some difficulty. In case of small seeded crops, it is very difficult to exercise the immature embryos, and aseptically located embryos which can be grown in a suitable medium under optimal conditions, and we have to provide the complex nutrient media also, because this is immature embryo. So obviously we have to provide the nutrient media with complex nutrients. And for adequate nutritional support of immature embryos, then this embryo endosperm transplant generally we will use. So we will see the what is this embryo endosperm transplant technique. Here in this picture you can see how we are doing. Here this is the hybrid species where we want the hybrid embryo, that is hybrid embryo we generated by crossing between two different species or two different genera of the wild plants. Here this is the ovule with the normal embryo. Here we are excising the embryo from the hybrid embryo, and this hybrid embryo will be placed in the normal endosperm by removing the normal embryo. This is the embryo endosperm transplant. The hybrid embryo from the ovule in which endosperm development has failed, that is taken out by the excise. Then another normally developed ovule, here we can see with the endosperm enclosing the embryo, that has to be chosen. And here endosperm is normal, but here endosperm has failed, here there is no endosperm. So for endosperm requirement purpose, we are transferring this hybrid embryo to the normal endosperm, and this ovule is dissected and normal embryo is pressed out. Here from here we are taking out the normal embryo, and it leaves a only endosperm with a exit hole. And now the hybrid embryo that can be inserted into this normal endosperm through its exit hole, which results in the embryo endosperm transplant that can be directly cultured on the nutrient suitable medium. By using embryo endosperm transplant, many interspecific and intergeneric plants can be raised. And third one is the ovule culture. In case of small seeded species, it is very difficult to exercise the immature embryos. So to prevent the embryo damage during the excision process, we can follow the ovule culture, that is we are exercising the ovule from the ovary. Ovule culture, that is we are growing the ovule culture or ovule embryo culture. As with the embryo culture, ovaries will be collected prior to the time at which embryo abortion is thought to occur, because we have to collect the embryos before the embryo abortion could occur. So the ovary is surface sterilized and ovules removed from this ovary, they are placed on the directly on the culture. And this ovule culture also may be very easy or may be very difficult, because where it is very easy, because in case of large seeded species, where it is very difficult in case of small seeded species, and it is very time consuming. Then coming to the other one, that is ovary culture. In ovary or pod culture, the entire ovary is placed into the culture. It is very easy because we are exercising the entire ovary, and we have to do surface sterilization process to remove the external contaminants which are present on the ovary. While placing the ovary, we have to keep the pedicel, because the pedicel used to be placed in the nutrient medium, because it will helps in the absorption of the nutrients from the this culture medium to the ovary, and from ovary inside the embryo. At the end of the experiment, seeds used to be removed from the fruit which are developed in the culture, and that seeds will be utilized for growing the viable plants. This is the embryo culture and procedure of embryo culture. So we learned about the different procedures, then what embryo abortion, that is how embryo abortion will occur, that is pre-fertilization barriers and post-fertilization barriers, and how to overcome these pre-fertilization barriers and how to overcome post-fertilization barriers, and what is the embryo culture, what is the embryo and endosperm transplant, and what is the ovule culture, and what is the ovary culture. I hope so you enjoyed this lecture, and please subscribe to my channel for the more videos which will encourage me to upload more videos. And if you feel that this lecture is very useful to you, please share with your friends also. Thank you for your keen interest in this video, and remember to like and subscribe for more videos. I wish you a great success, health, prosperity and happiness. Thank you one and all, and we will meet again with another video.